Inside every single one of your cells, there's a logistics network that's frankly more complex and efficient than any shipping company on the planet. Today, we're going to dive into two of its most critical players, the Golgi apparatus, think of it as the cell's master post office, and the lysosomes, which are like its high-tech recycling centers. So let's jump right in. All right, so here's the game plan. We're going to start at the Golgi, the cell's post office, and really see how it works as a molecular workshop for proteins and lipids. Then we'll get into the super elegant M6P pathway, which is basically the cell's internal zip code system. After that, we'll take a trip to the lysosome, the cellular recycling plant, and figure out how stuff even gets there. And finally, we'll look at what happens when this whole incredible trafficking system goes wrong, because the consequences are pretty serious. Okay, first stop, the Golgi apparatus. I want you to picture it as the central sorting facility and processing hub for the entire cell. I mean, this place is a non-stop, bustling center that's constantly receiving, modifying, and directing a massive stream of molecular cargo. And that post office analogy, it's honestly perfect. See, the Golgi doesn't make the initial packages. That's mostly the job of its next-door neighbor, the endoplasmic reticulum, or ER. What the Golgi does is receive that raw cargo, expertly tweak it, slap on a super precise shipping label, and then send it off to exactly where it needs to go. Now, this diagram here shows something absolutely crucial about the Golgi. It's not just some random stack of membranes. Nope. It's got a very clear direction, kind of like a one-way street for all this cellular cargo. Proteins and lipids show up in these little transport vesicles from the ER, and they enter at what's called the cis phase. As they journey through the different layers, they get processed and sorted, and then they're all packaged up and shipped out from the other side, the trans phase. So these flattened sacs you're seeing, they're called Cisternae, and they basically form a molecular assembly line. Think about it. Each layer, from the moment a molecule enters to the moment it leaves, has its own unique set of enzymes. It's just this incredibly efficient system where every single station does one specific job before passing the cargo along to the next. All right, so we've got the structure down, but what's actually happening inside this molecular workshop? Well, this is where the Golgi really shines. It's all about meticulously finishing and customizing every molecule that passes through its doors. So one of the Golgi's biggest jobs is customizing these complex sugar chains. They're called oligosaccharides that are attached to a ton of proteins. You can think of them like little flags that can totally change a protein's function or even where it's headed. These proteins arrive from the ER with a pretty basic set of flags, and then the Golgi's assembly line of enzymes gets to work, modifying them in this super precise step-by-step process you see right here. And hey, it's not just about proteins. The Golgi is an absolute powerhouse for lipid metabolism too. It takes this lipid called ceramide, which is made back in the ER, and completely transforms it. As you can see, it can turn it into sphingomyelin, which is a vital part of our cell membranes, or it can become a glycolipid, and that acts kind of like a cellular ID card, for recognition and signaling. Okay, so the Golgi has finished customizing all these molecules. Awesome. But now comes the really critical step, sorting. I mean, how does it make sure everything gets to the right place? Well, one of the most beautiful examples of this precision is what's called the mannose 6-phosphate pathway, or M6P for short. This is the cell system for creating a very specific shipping label. And here's where the system is just absolutely genius. For enzymes that are supposed to end up in the lysosome, the Golgi adds this special marker. A phosphate group gets attached to a mannose sugar. This M6P tag is, and I'm not exaggerating, the literal zip code for the lysosome. The cell has an enzyme that sees a unique structural patch on these specific proteins and just adds the tag. This makes sure they get separated from all the other cargo. It's just an incredibly elegant solution to what would otherwise be a massive logistical nightmare. But that leads to the next logical question, right? A shipping label is pretty useless if nobody reads it. So how on earth does the cell recognize this M6P tag and make sure the package it gets delivered to the lysosome and doesn't, accidentally get shipped out of the cell or something? Well, that very question brings us to our final destination, the lysosome. If the Golgi is the post office, then the lysosome is the cell's super sophisticated waste disposal and recycling center. It's this specialized little bubble filled with an entire arsenal of powerful digestive enzymes. So this slide right here highlights one of the lysosome's most incredible features. It has an intensely acidic interior. We're talking a pH of about 5.
To put that in perspective, that's roughly 100 times more acidic than the rest of the cell, which hangs out at a neutral pH of about 7.2. This isn't just some small difference. It's a fundamental state that the cell fiercely protects because it is absolutely essential for its function.
And how does it maintain this super acidic environment? It uses this thing called a proton pump, which is constantly funneling hydrogen ions inside using energy from ATP. And this creates a brilliant two-part safety system. First off, that acidic environment is what activates the digestive enzymes. They just flat out don't work at the neutral pH of the cell. And second, if a lysosome ever broke open, its enzymes will be instantly neutralized by the rest of the cell, which stops them from going on a rampage and digesting the cell from the inside out. Pretty smart, right?
So what exactly do these things do? Well, lysosomes are incredibly versatile. They break down stuff the cell brings in from the outside. That process is called endocytosis. And they also clean up the cell's own junk, worn out parts, in a process of self-eating called autophagy. Basically, if it's a major biological molecule, chances are the lysosome has the right tools to take it apart.
Okay, so now let's connect all the dots. We've seen how those digestive enzymes get their M6P address label back in the Golgi, but how do those enzymes and the stuff they're supposed to digest actually end up in the same place at the same time? Let's trace the whole pathway.
It all kicks off when the cell grabs some material from the outside. This cargo first lands in what's called an early endosome, which is like the first sorting station. From there, some of the stuff gets recycled back to the surface, but the rest, it continues on a one-way trip, maturing into a late endosome as it gets more and more acidic inside. And then finally, those transport vesicles from the Golgi, the ones carrying our M6P-tagged enzymes, they arrive and fuse, delivering their payload and creating a fully functional lysosome. This diagram just lays out the beautiful choreography of the whole thing. You've got two completely separate streams of traffic, right? One is bringing in material from the outside, and the other is delivering the digestive machinery from the Golgi. And they are both destined to meet at the late endosome, which then turns into a fully armed lysosome. It is just an incredible example of precision timing and cellular teamwork.
So far, we've seen this amazing system working just perfectly. But with this much complexity, it also means there are a lot of places where things can go wrong. And when these intricate trafficking pathways break down, the consequences for our health can be absolutely devastating. And this brings us to a whole class of over 30 genetic conditions known as lysosomal storage disease. What happens is the gene for just one single lysosomal enzyme is faulty. Without that specific enzyme, its target molecule can't be broken down, so it just builds up and up and up, causing the lysosome to swell and eventually mess with the entire cell's function. A classic example of this is Gaucher disease. It's caused by a deficiency in an enzyme that's supposed to break down a specific kind of lipid. So this undigested lipid just accumulates in the lysosomes of immune cells, and that leads to symptoms that affect the spleen, the liver, and even the bones. It's really a stark reminder of how one tiny molecular error can have massive, body-wide consequences. So what's the big takeaway here? Well, the fact that a single misplaced shipping label or just one faulty enzyme can cause this whole cascade of problems, leading to serious disease, it's just staggering when you think about it. It's such a powerful reminder that our health depends on the flawless execution of this microscopic logistics network, something that's happening trillions of times a second inside every one of us. It really shows you that every single part, no matter how small it seems, is absolutely essential.